From the Library of Congress in Washington, D.C.
This session is going to be called The Emergence of Life in the Lab. The last session we learned about how to get clues to the origin of life by going to lots of interesting and difficult to get to places like the bottom of the ocean. And earlier someone had mentioned about the recent discovery of fossils, which are maybe the oldest fossils of life on Earth, dating back over 3.7 billion years, which come from Greenland. These are tough places to get to, and you get little clues, sometimes very indirect clues. If you're looking at things that are alive today, you're not really looking at life as it began. And if you're looking at fossils, you're looking at these very kind of faint shadows of life that once was. So, another way to think about the origin of life, another way to study it is to say, hey, it's life and it's made up of molecules, so let's just put them together and test them out and see if we can make life, or if we can carry out some of the reactions we think were important for making life. And let's just do it in the nice, comfortable confines of a lab where you can get lots of coffee when you need it and everything's great. Not that easy, actually, either. And so we're going to be hearing from a scientist who does this kind of research, as well as a historian and an anthropologist, about sort of the culture of life in the lab. And so we'll start with Steve Benner. Steve is a distinguished fellow in the Westheimer Institute at the Foundations for Applied Molecular Evolution. And he has taught at Harvard and other places as well. I've had the privilege of writing about Steve's work several times over the past few years. I wrote a piece in 2013 for the New York Times about some of his musings about whether life is more likely to have originated here on Earth or have started on Mars and come here. So if you just Google our names in the New York Times, that should pop up. And I remember, I just wanted to recall, like, when I first met Steve, which was a while ago. It may have been, gosh, maybe 15 years ago or something. I was going to do a profile of Jack Szostak, who's also a very important person in the origin of life research, who's building these kind of protocells. And Steve was visiting, and they were just hanging out in a coffee shop, and they just said, Jack just said, just meet us here. So I sit down and am introduced to Steve. I've never met before. And he says, so what do you think? How much money do you think it would take to set up a lab to be able to invent life from scratch? Like, how much money, given what we know, just what do you think? And I just thought about it and said, $20 million? And he just paused and he thought about it. It's like, yeah, that sounds about right. So that gives you a sense of how Steve thinks. And without...
This is on. I guess it is on. And this is quite a diverse group, and I'm going to start first by talking to those of you whose only interaction with science was in a bad 8th grade middle school science course, where you were taught about the scientific method, which had questions and hypotheses and observations and tidily constructed experiments that tested the mechanism, the hypothesis, or not the hypothesis, but really missed all the basic issues. And so, I've given up judging science fair projects, but you can actually look at some of the problems that you have. Like, here's a guy who's got a poster who wants to know why chicks dig me. You'll notice there's a hypothesis. Remember, all these posters in a science fair have to state a hypothesis and a background and all the rest of it. But rarely is the language discussed as to what language will be used to answer the question. This is certainly a problem with the origins of life. What do you expect? A mathematical formula? Do you expect a chemical reaction? Do you expect a geological model? And of course, for any of you who have a small child and you say, chicks dig me because I have nice hair, the question is, but why do nice hair have chicks dig me. And of course, if you're a good Darwinian, at the end of the day, you say that, well, chicks dig this nice gentleman because they think that his DNA paired with theirs would give their offspring the highest chances of surviving itself, getting married and having children. But, you know, I mean, here's a great guy who had an interesting question. Will there be minorities in heaven? That's not his question. His question is there a heaven? And then some science fair advisor insisted on a purpose, hypothesis, variable format. And so he had to come up with a new poster, which is unfortunate. Now, simple questions are easy to answer, like this one: how long will a dead body float? Right? And of course, these are answers can also be put to practical use. Here's a science fair project called Getting Away with It: Proper Corpse Disposal, in case you're interested in knowing this. But keep in mind, this is the simple questions that are easy to answer by this hypothesis-based direction, but not the big ones. And unfortunately, the big questions are the interesting ones. So I got tired of my kid being abused by 8th grade science fair project leaders, so I wrote this book, Life, the Universe, and the Scientific Method, a copy of which I have here, which I will autograph for you if anybody wants one. Basically, science is not hypothesis testing. It's a back and forth between two sides, or maybe 5 sides, or maybe even more. Everybody's trying to place the burden of proof on somebody else, right? Nothing is proven in science. Nothing actually is disproven in science. Karl Popper and our education of scientists notwithstanding. It's an operational science as the back and forth can be very well organized and people understand who has the burden of proof. But when you say that metabolism comes before biopolymers, this is metabolism first or DNA first, that becomes a confused burden of proof to assign. Or life arose at deep sea thermal vents, which is what I put in just after hearing the last talk. These are not so much yes or no questions as what do you really mean? And given the state of the field, discussions are often, you'll see this, semantically confused without understanding where the burden is. And really, we have to decide who must show what in order for this field to advance. Now, we've been involved in what we call the 4 paths to understand origins. It's a little Chinese, a little Confucianism. But, you know, the northern path is where we try to build life from the pieces, the bottom. The eastern path is the path which we discussed a moment ago about where we try to construct our own life forms. Obviously, we are desperately trying to get married to get us an alien from Mars. If we can just get another species from Mars who arose independently of ours, we would have a great insight to this. But I'd like to start with the bottom one, which actually somewhat makes the most sense, right? If you have a biosphere full of life, you know, you might as well try to look at that biosphere and see how it might have worked. And keep in mind, your problem is that, as we have shown on this green tree, there is a natural history which relates all the organisms that we know to each other by common molecular structures. Now, that's a bit circular because the way we look for organisms on Earth is by looking for those molecular structures. So it is conceivable that there is a shadow biosphere, that's Carol Cleland's term, which might be under our feet, which uses a different biochemistry, which we don't see because we don't know how to look for it. And in some sense, Carl Woese, who you've already seen a picture on the slide, has perhaps discovered one of those many years, 50 years ago about now. But working backwards in time, we are using sort of a process, and this process is a principle of minimal motion. We use it, for example, when we construct Indo-European languages. This is just the Indo-European language, Slavic, Germanic, Romance languages, Celtic, which has the word for snow. It's present in all of those languages. Of course, in some cases, it's quite recognizable. So German, it's Schnee, and Slavic, it's Schneg. In French, of course, the Romance languages decided that SN was hard to pronounce, so they dropped the SN, so you have just the words beginning with N, like neige in Spanish, or neige in French. What's important, of course, is now you can actually look at the structures and you see them in common from all the descendants. And the principle of minimal motion, some people call it maximum parsimony, is to say that if I assume that languages change as little as possible, I will try to construct the ancient word for snow and the last common ancestor of all of the modern Indo-European languages with a little, smallest amount of phonetic or change. And to do that, you can come up with the word schnig, which is written at the top of this chart as what you would say to an Indo-European if you were to encounter him or her in the street and wanted to talk about snow. Now, if you put these words into natural context, however, they get more than just that, right? And the natural context, all of a sudden you realize that Indo-European has more words, and it tells you not just about what you would say to the Indo-European if you encounter him or her on the street, but it also tells you something about the Indo-European society. Specifically, it lived in a place where it snowed, which rules out, say, Zaire. But, you know, the Reconstruction Indo-European language does not have a word for gold. It does not have a word for silver. It does not have a word for, you know, tax-exempt mutual bonds. But it does have words for gold, for dogs and for sheep and for oxen and for pigs. They have grain. They can put them in vehicles which have wheels, and they can count to 100. And they're all, we know that by looking at the context of the modern languages set into a model of how you infer ancient ancestral structures from the structures of descendant words. So we can do the same thing now by replacing the phonetic letters by molecular structures. And let me just do something that's important, because I'm a chemist, and you've heard Wally Gilbert, he mentioned he was a physicist turned molecular biologist. We chemists look at molecules atom by atom, right? We don't, and we look down upon, and you'll probably get the tension in my voice. We resent personally that Wally Gilbert, a physicist turned molecular biologist who looks at structures only letters at a time, does not look at atoms, but nonetheless wins the Nobel Prize in chemistry. That gets us real tense. But let me go back, and I'm not sure who has looked at their vitamin pills. Please raise your hands if you actually study. So if you look at this, you'll notice that there's a whole bunch of stuff in here, and what these generally are what we'll call enzyme cofactors. You've heard enzymes a couple minutes ago. These are the catalysts that catalyze transformations. I've just put down here a structure of ATP. ATP is well known as being a very common molecule involved in phosphate transfer, phosphorylation, but also energy economy of cells. And I've structured it to show you that there's a piece of RNA attached to it. And the black part is the phosphate that's actually been transferred. The orange part is RNA. And all modern organisms, at least that we know of on Earth, have ATP in them. And so we infer from that, and the principle of minimal motion, that the last universal common ancestor, which charmingly has the name Luca, also had ATP. But of course, it's more than that. If the last universal common ancestor had ATP, it was also phosphorylating. Now that's a leap that requires a logical premise. It's sort of the same kind of logic that if I gave you a T-Rex tooth, right, and it looks like it's ideal for chewing meat, then you infer that the T-Rex was a meat eater. That's a deep Darwinian statement, actually. But the bottom line is that if all descendants of Luca have ATP, the ancestor Luca had ATP, all descendants use ATP to transfer phosphate, therefore the ancestor transferred phosphate. And if you go back to your vitamin pills, you'll notice that these are so important that there's niacin, which I have as the structure there. I'm thinking the mouse shows up on the screen, does it? We have, of course, thiamine. We have coenzyme A, which is you eat in the vitamin pills as pantothenic acid. All of these molecules end up in your metabolism with a reactive part in black and an RNA piece attached to it, doing nothing. And the reason why we know it's doing nothing is because for all of those structures, I put a little blue structure, which is the same molecular structure that can do exactly the same thing as the RNA cofactor can do, but it is not used very widely in enzymology. The ancestral state, Luca had RNA attached to everything. Now that leads to an obvious question. It was a question raised by Harold White, a chemist, University of Delaware, 1976. It was raised by Cornelius Visser, a chemist, and Richard Kellogg, a chemist, both in Groningen, Netherlands, in 1998. That paper in 1998 is how biotin's reactivity can be understood, biotin being one of the things you eat, in terms of the RNA world. We're 10 years ahead of the molecular biologists. I'm hoping Wally is not watching this because I'm going to get a long letter from him. We're 10 years ahead of the molecular biologists because we look at the chemistry, and of course that paper from 1970 is what persuaded me that the RNA world was so likely to be the case because it explains so much about the molecules that we have in our everyday life. Everybody with me? Cool. So it turns out that there's lots of other things. The RNA world, of course, as you've already heard today, is defined as an episode in natural history on Earth where RNA was the sole genetically encoded biocatalyst. It explains these RNA cofactors with RNA handles to bind RNA enzymes. Again, DNA later emerged for more stable genetics. You heard that a few minutes ago. And then RNA catalysts later emerged to make proteins. That structure in blue and orange is the ribosome. It's the machine that makes proteins, and I've color-coded it. So the orange is RNA. You've probably gotten the thread here. The blue is proteins slapped on the surface, which help out. But the ribosome, making proteins, is an RNA catalyst, and that is in addition to the structure of the RNA world cofactors. Now, for this to make sense, okay, RNA world must not only have had the cofactors, but also the reactions that use the cofactors. That is, the RNA world must have had a complex metabolism with redox metabolism, that is oxidation and reduction, because that's what niacin RNA cofactors do, and that's what flavin or riboflavin is what's in your bottle. Cofactors do clays and carbon-carbon bonding forming reactions. I won't go through all of the chemical jargon that describes the reactions that the RNA world was catalyzing, but if this theory, or if this explanation, if this why question for your 8th grade science fair posters to make any sense, it's got to be the case that not only did the RNA world have those cofactors, but it used them, and it must have used them in a complex metabolism. And we actually in 1989 wrote this paper with Andy Ellington, now at the University of Texas, The Modern Metabolism as a Palimpsest of the RNA world. And in 1993, in the book that you've already seen mentioned, we actually wrote 50 pages to write out all the details of what that metabolism looked like. And so you've heard already, this is the same thing now with a niacin cofactor. You heard a moment ago about Bill Martin's view. Bill Martin's bunch of people looking at organic biomolecules as if they're strings of letters, not looking at the individual atoms in them at all. And they have come up with a conclusion that completely denies this. Now, in our view, that's wrong. Their view, of course, we're wrong. So we can go back and forth trying to shift the burden of proof to each other as we duke it out in the bold alpha male world of modern science. Okay? Fair enough. So that's a problem for us, because working backwards in time is going to fail to get us anywhere near origins, right? If you believe Bill Martin, you don't agree with us, but remember, if you believe Bill, you can't explain the RNA cofactors. You can explain the RNA cofactors. This is what I think I like about my Willard Van Orman Quine moments. I mean, I don't know if for those of you who are into philosophy, Quine basically made a remark about Karl Popper's statement that science was associated with disprovable hypotheses. And Quine's comment was, no, you can't disprove anything either, because there's always these auxiliary hypotheses. You know, the experiment failed, disproves the theory? No. It proves that the machine was broken, right? I mean, there's all sorts of stuff. But one of the nice things that Quine does talk about is these packages of logical statements that you choose. So Martin is right, can't explain RNA cofactors. Can't explain RNA cofactors, then Martin is wrong. And so what emerges, we have to really be forced now to go into the north quadrant of this plot. We actually have to worry about this model. And of course, what going backwards in time has done for us is not nothing. It's done something for us. It does suggest an origins model that must be dependent on RNA. That's what that story has done for us. The story is, and there's Jean-Luc Picard with Q, who is flitting in and out of the continuum, taking Jean-Luc Picard precisely at the time where life originated on Earth. The argument is that RNA emerged able to catalyze template-directed synthesis of RNA with errors, but where the errors are replicable. If you're a good, hardcore molecular Darwinist, that's all you need to have this process of having kids with mutants and having the better ones survive and the worse ones die, produce all of the behaviors that we value in life. Now, the first person I know who did this was Alex Rich in 1962, and frankly, he wrote that out much better than Woese, Oracle, or Crick. It was a very clear statement. And Alex died last year. He's a great hero of mine, but he was also very persuasive. But I did not know about it until I gave a seminar where he was in the audience, where I cited Watson, Crick, and Crick, Oracle, and Woese. So now there are molecules. Phil Holliger, actually a former student in my lab. For those of you who've been in my lab, you'll know that I will take credit for everything you do ever after. Actually, I had absolutely nothing to do with this. He's at MRC in Cambridge. He made an RNA molecule that is actually able to catalyze the template-directed synthesis of RNA with errors, but they're replicable. So, are we done? Done? and done, right? Well, not so fast. This is a detailed hypothesis. I would prefer the word model. You can say that the burden of proof is shifted, and I can insist that others now disprove the hypothesis. Well, good luck with that. And so the question now comes back to us, how can this model meet a convincing burden of proof where the convincing people, the people needing conviction, are creationists as well as our professional colleagues in molecular biology as well as, of course, the chemists. And that's a big problem because we should be able to assemble in the modern lab components of RNA from prebiotic precursors, especially since we're not operating under the constraints of a prebiotic soup, right? I mean, compared to the prebiotic earth, modern molecular biologists have the power of God, right? We ought to be able to go back and do this trivially. And so, but keep in mind, I mean, I'm going to pass these samples around because this is what we're looking for. This is a stromatolite. This is 3.4 billion years old. It's not the 3.7 that has just recently been. And I expect these to come back. I do, however, want you, this looks like an honest crowd, yes? But I just want to, there's Cambrian explosion. That's actually something that you might say looks like you. That's the origin of life, sort of, I guess. I mean, but there's twice as much time between the origin of Earth and this guy as there is between this guy and you. So you should keep that in mind when you were. So here, pass these around and then drop them off in the back when you're done. Set them down carefully. Oh yeah, there, yes. Well, so we ought to be able to do it, and what's happened when people have tried, and it's about 50 years of failure. So what was one doing? Now, you've already heard this before. What you want to do is you want to, in this business, follow the paradoxes. You want to understand what it is that is so goddamn difficult. And then you want to go back and look at that with intense focus, one at a time, because there will be one goddamn thing after another as you go through this. So I put out 5 paradoxes for you. One's the so-called tar paradox, which is if you give enough energy to organic matter, which happens to not have access to Darwinian evolution, you don't get life, you get tar, and it's a well-validated observation. And you probably have validated it yourself in the kitchen if you've left the oven on too long, right? You get tar. The phosphate paradox, we'll talk about in a minute. Phosphate is the linking groups in RNA. It's hard to get phosphate in the presence of calcium because you get something that looks like this. This is apatite, it's calcium phosphate. The other thing is also possible is your bones, which is, so I can spend, lovely, these are lovely stones, by the way. Yes, I don't mean to disrupt your. So the third paradox we'll talk about is the water paradox. We say that life requires water, but water is a very corrosive solvent, and it basically destroys all the biopolymers and a good deal of the other molecules, including a lot of the cofactors that I mentioned. Next, suppose you get over that. You get the tar, manage to evade tar, prevent the organic molecules from devolving to get asphalt. You manage to get the phosphate, you manage to assemble RNA, you manage to keep it. The next paradox that you will encounter is that once you've made the RNA, it's not stable enough to accumulate. And so you have to worry about recovering the RNA. And finally, even if you can keep RNA, it's far easier to get RNA that destroys RNA as a catalyst. We can do that, roughly 1 molecule in 10 to the 14th of RNA molecules as a catalyst able to destroy RNA. Much more easy to get stuff that destroys than to create. We don't know the actual likelihood of the odds for making RNA, but it's somewhere in the 10 to the 30s. So let me go through and just tell you a little bit about what goes on here when you focus on these paradoxes. And I'll start with the tar paradox, which is well validated. It's known in sugars. There's sucrose. Who's here grown rock candy? Raise your hand. Someone. That was in 7th grade, right? So that's a great, but if you heat it a little bit, you'll get caramelization, right? You'll see, and you're looking at it, right? More heat, more caramel, and this is well known to you, and unfortunately, that's a sugar, but RNA has a sugar component, which is unfortunately R. And since Miller's experiment was named, I want to point out the power of rhetoric in science. Left is Stanley himself, the great man who arguably founded modern prebiotic chemistry with his machine in a press photo. The right picture shows what it actually looks like. What Miller showed, that if you put energy into organics without Darwinism, you get tar. So why do you get tar? Well, the answer is because of for the chemists, I'll talk to you, we talk about enolization and aldol reactions. For the non-chemists, I just want you to look for C double bond O, a carbon joined by 2 bonds to an oxygen, because that can do these reactions, enolization, which is shown here, and aldol addition, which is also shown here, that give you complexity, which looks like that. And of course, I've only put part of the complexity in, and we've worked out all these reactions. One of the problems with ribose is it also has a C double bond O unit, and therefore it can enolize, there's that reaction, and react with itself, and it can form asphalt. In fact, it's so bad that in 1995, Stanley Miller wrote a paper, which I've written on this slide, where he says, and I'm quoting now, stability considerations preclude the use of ribose and other sugars as prebiotic agents. They fall apart, ribose falls apart. It follows that ribose and any other sugars were not components of the first genetic material, and that's a real problem for the RNA world. Monica, if there's no R, ribose, there's no RNA. So, you know, we've been now for 15 years making this plea for mineralogy, partly because when I was a kid, I was a rock hound. Life did not begin in a Pyrex baking pan or in a Pyrex beaker, for that matter. It began in a mineral environment, and I'm going to actually pull up, here's a wonderful mineral for you, actually hard to see. These are tourmalines. It's probably better on the picture. It's even better down the street in the jewelry store. Tourmalines contain the element boron, and boron with oxygen gives borate, and borate is something that gets excluded from rock-forming minerals and comes to the surface, and it washes out into alkaline streams. You heard serpentinization. That's coming from peridote. There's an example of peridote, which you can actually see the serpentization. Borate has a magical property. It binds to 2 adjacent OH hydroxyl groups. Ribose has adjacent OH hydroxyl groups. Once borate binds, it causes the whole molecule to cyclize and get rid of the C double bond O group. The C double bond O group is what gives you so much trouble in prebiotic chemistry. It also happens to guide the reactions of carbohydrates. And in fact, if I just go back to the picture I showed you before, borate constrains the reactivity to just that little region of the plot. Wonderful. So there's a lot of stories on the tar paradox. That's just one. But let me now move on to the phosphate paradox, which comes from the fact that it's hard to get high concentrations of phosphate in the presence of calcium, which precipitates as apatite, which is now circulating throughout the room. And apatite does not phosphorylate ribose or sugars, and in fact does not allow itself to easily go into the backbone of RNA. But, you know, here is a shout out for astrobiology, because astrobiology as an institute, the NASA Astrobiology Institute, went out of its way to create what we call multidisciplinarity. But then they did so by having a virtual institute. They did not require universities to move people from place to place and build a building to house them. Multidisciplinarity is certainly accessible by the internet. And so you got to really enjoy browsing geological reports. I don't know who, the people who are geologists, you sit back, and I'm sitting in the sunroom one day, you know, doing the crossword, and then I pick up this geology report, and I notice here gypsum occurs within the sequences. These are geological sequences containing Lüneburgite. Well, okay, what the hell is gypsum? What the hell is Lüneburgite? Well, gypsum is calcium sulfate. Mineralogists have this name just to keep the club small. Lüneburgite is a magnesium borophosphate. And the fact that they occur together has significance. So what does it mean if gypsum, which is a combination of magenta calcium and yellow sulfate, and Lüneburgite, which is a red magnesium, a blue boron, and a green phosphate, occur together? Well, as I mentioned, the phosphate problem in the RNA first model has to do with the need for a lot of phosphate to make RNA, and phosphate, green, and calcium, magenta, generally precipitate as magenta green apatite. But in the presence of borate, what that geological report told us was that no, if you actually have borate present, blue, it will sequester the green phosphate away from the magenta green apatite. And in fact, if you, instead of having the 2 boxes on the left where calcium and phosphate precipitate to get useless apatite, and magnesium sulfate precipitate to get useless epsonite, with borate present, phosphate, borate, and magnesium go together to give you Lüneburgite, and calcium and sulfate are separated. You've rescued the phosphate from the calcium. And this also happens in the laboratory. So, different question. Right? A minute ago, we used boron to stabilize organics to prevent them from forming tar. Here, we're now using boron to sequester phosphate away from calcium so that it doesn't get lost in a useless mineral. Two questions, same answer. It's a wonderful thing. And it turns out that Lüneburgite does a wonderful job not only in making the phosphate available, but once the nucleoside, which I have drawn up in the right-hand corner of that slide, extracts the borate out of the mineral, it then gets itself phosphorylated. And more to the point, it actually gets phosphorylated just at one place, the 5' position, which is where you want it to. Now, by the way, we're talking about continuity, right? You don't want to have a step happen and then have an intelligent designer have to come in and take the material that was made in the first step of prebiotic chemistry, redo it, purify it, then put it into the second step. If you can get all these steps working together, okay, you're in great shape. I've actually put a structure up here which is a nucleoside. It's a building block for RNA. You might ask, where do nucleosides come from? In a desert, about 4 months ago, Tom Carell actually proposed a route for this, which I've shown here. So what we're doing here is now building what will emphasize the weakness of this model. The weakness of this model is it's still discontinuous, which gets us from gases like carbon dioxide and hydrogen, hydrogen oxide and dinitrogen and methane, to formaldehyde. I wish that Jim had been here because he actually wrote the distinctive review of this. We then get to the other processes in stepwise form, going from atmospheric to, in this case, ribose, nucleosides, phosphates. How are we doing? A lot of chemistry. Obviously, the next question is, even if you make RNA, how do you get it to accumulate? Well, it turns out that minerals in a desert also bind to the RNA and stabilize it. This is work that's just appearing from Elisa Biondi in our group, where she is absorbing RNA that is made onto the support. And so next question is, even if you can keep RNA, it's far easier to destroy RNA than it is. It's far easier to get RNA that destroys RNA than it is to get RNA that makes RNA. How much time do I have? I'm looking at 3 minutes, but we started quite late, right? So. But let me just spend the 3 minutes now just to give you the final paradox, right? Keep in mind that RNA is a bad reservoir of catalytic activity. The reason it took so long for Tom Cech and Sid Altman to win the Nobel Prize finding catalysis out of RNA was it's not a very good molecule. And that's because it has only 4 building blocks and not a lot of functional groups like you have on amino acids and proteins, which is what makes proteins so powerful as catalysts. However, if you again do what I showed you before, looking at the modern life forms and going backwards in time, you'll discover that LUCA does not just have RNA. It has RNA with these magenta side chains attached to it, which are functional groups. In fact, some of them are very close to the functional groups that are found in proteins. And so maybe at some point, LUCA decided, maybe before it invented translation, maybe Terran life may have tried to improve the catalytic power of RNA by adding functionality to the standard basis. Now, of course, you and I descended from an organism which, as Mary pointed out, was the winners who did not do that, but maybe we as synthetic biologists can create an alternative path and do what natural history did not do on Earth. And so this is now not backwards in time, it's not forwards in time, it's the east quadrant, where we're actually going to take DNA, which I'm just going to use the Lego structures to illustrate how DNA works with a big and a small base pair with prongs and holes. We're going to complete this Lego set, so we actually have all possible combinations of big bases and small bases and prongs and acceptors. There's the chemistry. There's some more chemistry. We're going to add functional groups to them, and we're going to have our own little private, personal, evolving Darwinian system, following a path that Terran life could have taken, but did not. And the reason why we can do this is because we are gods compared to the prebiotic Earth. So I won't describe this because we're out of time. I will just say that we are able to apply Darwinism. Keep in mind that we do pay the bills in our laboratory by binding to cancer cells. Why? Because that's a much more important thing to bind to. But once you have just 4 letters in your nucleic acid alphabet and you try to evolve from that 4-letter alphabet DNA or RNA molecules that bind to cancer cells but not to non-cancer cells, that's a lack of specificity. It's hard to do. But if you add letters to the genetic alphabet, you get those that in fact bind quite well. So there is the paradox is that you can go back by focusing on these individual details, bring in the geological environment and the minerals. Well, you can ask, were these minerals and deserts present on early Earth? Bob Hazen and I have had a long discussion of this. Were deserts available on early Earth where you could have had these things evaporate? A similar discussion. What's nice is that the minutes that our geologists look for Archean borate, they discover it. So Lüneburgite, for example, is unstable to metamorphosis. It gives monazite, it gives apatite, it gives tourmaline, but you can find all of those minerals in 3.8 billion-year-old geometric metasedimentary rocks from Greenland, which Carl mentioned. Are deserts available on early Earth? Joe Kirschvink says no. Water inventory much too high for there to be any dry land. So Joe has suggested the Kirschvink model, which was that all you got to do is go to Mars, which has always had less water. The logic tree, of course, is you've got to decide now whether the burden of proof has been met. RNA first, yes or no. If it's no, you have a different set of problems. Benner's stupid chemistry, if the answer is no, my response is no, get your own goddamn chemistry. If it's yes, you need an oxidized desert, minerals in a desert. Can we have those on Mars? You've got to decide. If the answer is no, then Mars, you can have them on Earth, then you have to decide. If the answer is no, well, then Mars is an option. If not, you can actually have the discontinuous model occurring on Earth. Well, with that, let me stop. I've said too much chemistry, that's for sure, but I think the chemistry is very important, and we'll go on to the next.
For the interest of time, we're going to, we can take questions at the end from everybody. We're going to do our best to catch up a bit. We may run a little bit late at the end of this session, but we'll do our best. And I guess you can see why whenever I interview Steve, I make a recording. I can go back and reflect. Anyway, our next speaker will be Luis Campos, who is an associate professor of the history of science at the University of New Mexico and the secretary-elect of the History of Science Society, and will be soon be the next Baruch Blumberg NASA Library of Congress Chair of Astrobiology here. So, Luis, please come join us.
Good morning. Soon, we are confident life will appear in a test tube, and our world will never be the same. Although this passage comes from a book published in 2000, the idea that it's merely a matter of time before life can be created in the test tube is hardly a new one, and it's come in many different flavors at many different times. In 1837, for instance, by running an electric current through a laboratory apparatus, the experimenter Andrew Cross was astonished to discover, crawling out one day, a species of Acarus mite, and a heretofore unknown one at that. These experiments in electrobiology proved to be remarkably popular. Although his laboratory setup was later disparaged by critics as an obstetrico-galvanic apparatus, his efforts were described by the historian James Secord as the most famous experiment of the first half of the 19th century. For most readers, he concluded the creation of life was an experimental fact by the end of January 1837. I want to start off today by suggesting that claims to have artificially synthesized new forms of life in the laboratory are not only oddly recurring, but a characteristic and a regular feature of the history of modern biology. From late 19th century announcements of Luther Burbank's new creations in fruits and flowers, up to the promises of contemporary synthetic biology today. Far from being solely sensational or merely curiosities, these claims to have synthesized new forms of life, when we examine them collectively and historically, all share a similar structure. The most recent of these are all cast in the tense of the future perfect, that we've not yet created life, but we will have done so someday soon. The creation of life in the test tube is always imminent, but it's also always proved to be deferred as our understanding of life shifts from cell to protein to gene to DNA and so on. What we once thought was the next obvious step that would finally reveal the secret of life with the creation of life in the test tube as the proof of principle is always deferred. As we heard from Matt this morning, we're still a long way from the first functioning protocells. So understanding the structure, the history of these claims may give us some valuable insights into the prospects for any future synthesis of life. And more to our point here, I want to suggest we can learn something from this history that might help us understand our current position in astrobiology in a new way. After all, we haven't found life yet, but we will someday, or at least we hope to. And the discovery of life in other elsewheres is always imminent. So as we hope to realize a more perfect future, we should see what we can gain from analyzing these claims of the future perfect in synthesizing life and in discovering it beyond the Earth. And since I'm a historian, the way that I do this is by thinking through some cases. And so I'd like to share some cases from my research in this very earlier history of origins of life research and history of genetics. So history is not in the prediction business, I'm sorry to say, but I hope to show it can be good to think with here. So 60 odd years after Cross's electrobiological experiments brings us to another set of experiments that were also described as sensational. And these involved the new phenomena of radioactivity in the hands of a curious Cavendish laboratory physicist, and again, it's the physicist there, J. Butler Burke. Burke, like Cross, was something of a one-hit wonder when it came to great experiments. His 15 minutes of fame came in the summer of 1905. Plunk a bit of radium chloride or bromide from an ordinary test tube into sterilized beef gelatin, or bouillon, Burke said, nothing more than beef tea, as one observer reported, and the result was strange growths in the medium. Over the course of their existence, these growths were found to grow in the dark, decay in sunlight, multiply by division, and even, it appeared, show the structure of the nucleus of a cell with the attendant phenomena of karyokinesis, the movement of chromosomes. Something queer has happened in his bouillon, reported some observers. Here's how this got reported fairly early on. I took the story on the right-hand column there and blew it up so you could read it. So unlike some of his contemporaries who were engaged in projects aimed at mimicking some fundamental life processes in inorganic media, he didn't just want to mimic life, he wanted to get at its underlying features. And he held that his experiments showed a kind of physical metabolism, as he called it, somewhere between accretion and assimilation. It was clear to him that what he had produced were not bacteria, or recognizably life as we know it, they were something else altogether. Neither radium nor microbe, Burke took these radiobes to be a new transitional form, not quite living, but exhibiting many of the characteristics essential to life here. Burke's experiments in artificial life, and he uses that term as his own, and the claims he made for it were quite modest when compared with the enormous stir that his experiments had aroused. He published his findings in Nature in 1905, but time and again the newspapers were reporting that Burke was getting at the secret of life. According to the Daily Chronicle, these radiobes existed in the critical state of matter between the mineral and the vegetable, in fact on the borderline between the living and the dead. According to the New York Times, these creatures were on the frontiers of life, where they tremble between the inertia of inanimate existence and the strange throb of incipient vitality. They were the prehistoric ancestor of the microbe, the Times reported. Another newspaper reported that Burke had succeeded in producing cultures bearing all the semblance of vitality. Radium, with its half-lives, had produced radiobes that were themselves half alive. Burke himself noted that the radiobes appear to possess many of the qualities and properties that enable them to be placed in the borderland, so to speak, between crystals and bacteria, organisms in the sense in which we've employed the word, and possibly the missing link between the animate and the inanimate. Although they, quote, obviously lie altogether outside the beaten track of living things, yet they were, he said, within the realm of biology. The question of whether these microscopic forms described are or are not living things, he says, depends altogether on what our conception of life is and upon how broad or narrow is the definition that we're willing to accept of the phenomena of vital processes. So while Cross, with his electrical mites, had produced obviously living things, just what Burke had produced was eminently contestable, and his discovery was described as one that has provoked more discussion, perhaps, than any event in the history of science since the publication of The Origin of Species. Had he produced living things or only lifelike things? Any claim to have produced life in the laboratory would be controversial by this point, an example of mostly discredited theories of spontaneous generation by around 1900. And as he himself noted, to expect to make a full-blown bacillus at the present day would not be more absurd than to try to manufacture a man. But to bring about certain phenomena by the action of radium that approached the properties of a living thing, that approximated life, was to be properly scientific and might even give insights into other realms in biology. Burke held, in fact, that his radiobes gave insight into a potential angle for research into the origin of life in the distant and remote past. In his 1906 book about his experiment, The Origin of Life, Its Physical Basis and Definition, he described how his radium-based approach, and radium was all the rage in 1904, meant that there was now experimental access to the question of the historical origin of life. There was a new way of reading what he called an obliterated page in the history of our planet. Simply put, the early Earth must have been more intensely radioactive, so there would have been more radium available to act upon the earthly bullion in such a manner. So maybe primordial soup is out, but this is even before the primordial soup, we have this bullion here. So this planetary conception for the origin of life was seen by a wider readership at the time as essential to understanding the place of life in the universe more generally. It's more than probable, the Daily Chronicle said, that radium is one of the ABCs of the stepping stones to a law of cosmic life. So these are the so-called radiobes. And these are the images that he publishes in his book. And our standards for presenting images have changed quite a bit since 1905, but I thought you might like to see what it is that he had presented, that he showed as evidence of these growths that grew and subdivided. So in producing a half-living thing that possessed, he said, n-1 of the n properties of living bacilli, Burke was broadening the understanding of what it would mean to study the emergence of life on Earth or in the universe. Now, he's not the best role model for scientific success, and he ended up going a little bit off the deep end. But by 1931, he had written a book on the subject entitled, perfectly for our purposes here, The Emergence of Life. Now, I wouldn't exactly recommend that you read this book. It's rather far from the mainstream even for his day. But in this little case study of his early experiments that we're seeing here is the very beginning of an experimental approach into the historical origin of life on the early Earth. And the question of the emergence of life here is already placed into what we might today call an astrobiological context. I find that fascinating and delightful and terribly relevant for us as we consider what we might learn from the history of attempts to create novel forms of life through laboratory experimentation and what we think we can come to know about the existence of life elsewhere in the universe. Let me move ahead with the story through some other figures in the history of attempts to create life in the test tube as part of our efforts to truly understand the nature of life. There are lots of investigators we might point to in this time, and the sheer number of attempted and failed attempts at artificial life led one author in Scientific American in 1911 to say, "From time to time, we're informed that the riddle of the universe has been solved by the artificial production of life from non-living materials. But each time we wondered how it was done for a few days, and then we find out it wasn't done at all. The riddle is still there. The expression artificial life has also been used in recent years for two entirely different sets of ideas. So one of them was Burke's approach, the production of life that never before existed. And the other one had to do with altering the characteristics or the distinctive properties of living matter. At the top of this list was Jacques L'Herb's recent discovery of artificial parthenogenesis, that by changing the concentration of salt in solution surrounding a sea urchin egg, he was able to cause the development of an embryo without fertilization. The next generation came into existence without a father. This most vital discovery in the history of physiology, almost the manufacture of life in the laboratory, as L'Herb was reported to have said, meant that we have drawn a great step nearer to the chemical theory of life and may see ahead of us a day when a scientist experimenting with chemicals in the test tube may see them unite and form a substance which shall live and move and reproduce itself. Now, for L'Herb, understanding a phenomenon meant being able to control that phenomenon. And so the ultimate control over life for him, what he called a technology of the living substance, was not only to be able to do with life as one willed, but eventually to be able to create it from scratch oneself in the test tube. So he wasn't trying to shock with these sorts of statements. It was just simply a concomitant of what he viewed as a thoroughgoing engineering approach to life. These sorts of exciting claims, however, about the newly discovered potentials of life and how we might engineer it, these claims have never been far from their publics. By 1938, Reinhard Beutner, in his Life's Beginning on the Earth, said that we must realize in all of these scientific activities there are certain rules that cannot be neglected. Scientists shouldn't draw suspicious conclusions from their findings, as for instance to say that moving oil drops represent a form of life. He said, if they violate this unwritten law, all of their results will be dumped into the wastebasket of disregard, even if real treasures are contained therein. Another unwritten law, he said, by which every scientist must abide, forbids the announcement in the lay press of unverified results that may give rise to general misconceptions. The public, of course, cannot possibly be informed on all details of every line of scientific research, hundreds of which are being pursued, and any announcement that artificial cells have been made may be hastily seized upon as the dawning of a new era. Alexander Oparin, with his influential early theory of coacervates as primitive cells, seems not to have heeded this advice. So rather than attempting a synthetic approach to the origin of life, he argued in 1934 for a more analytical deduction from postulated early conditions. He said, we're faced with this colossal problem of investigating each separate stage of the evolutionary process. We have to delve into the properties of proteins. We must learn the structure of colloidal organic systems, of enzymes, of protoplasmic organization. The road ahead is hard and long, but without doubt it leads to the ultimate knowledge of the nature of life. The artificial building or synthesis of living things is a very remote, but not an unattainable goal along this road. So as we near mid-century then, the process of conceptually distinguishing the spontaneous generation of life, the question of a historical origin of life, and efforts to produce life artificially or synthetically is nearly distinct. The first one is entirely out of court, the second one is of great interest in current research, and the third is the eventual but still distant and deferred goal. By 1953, of course, we have the famous Urey-Miller experiment we've heard about several times, another approach intended to give insights. And just as Oparin had acknowledged but ultimately deferred the synthetic goal, so Miller's experiment was also reported in the press as heralding, though not yet, bringing about the eventual artificial synthesis of life. And in the context of Herman J. Muller's important studies on the gene and a virologist's work on viruses, among others, their genes and viruses were increasingly seen as natural bridges of sorts between the living and the non-living. And at the same time, this increased study of real living things became increasingly connected to questions of how one might engineer such systems. So my point here is that evolutionary novelty and engineered novelty have never been very far apart. While J.B.S. Haldane believed that a self-contained organism will not be made for centuries, and N.W. Peary had provided some pretty good criticisms by mid-century of any attempt to produce life artificially, by 1964, Oparin had become more optimistic about the possibility. And by 1967, J.D. Bernal wrote, "If life once made itself, it must not be too difficult to make it again." And elsewhere, "But the final proof remains to be found, and that is by actually making what we're talking about." Or as David Grinspoon has said even more recently, if we succeed in creating life from scratch in the lab, we may still not know how it actually happened historically, but our belief that it did happen will gain several notches in credibility. So such efforts in the laboratory may well give us insights into how life has emerged in other times and at other places. So this brings us back then to the world of astrobiology. Attempts to produce life in the test tube have for decades been closely associated with the quest to understand how life may have emerged on the early Earth or in the universe. And this was certainly the case in the early years of exobiology. James Strick and Stephen Dick tell the story of an exobiologist entering the new lab at NASA Ames and being, quote, "a bit horrified to find when I first got there a secretary answering the telephone with life synthesis." So from this, I want to suggest that the history of claims for the production of life in vitro and its discovery in astro, both the engineering of new life forms and the potential discovery of new life forms elsewhere in the universe, share common features. And it might be useful to think through the history of the one in order to help understand our position in the other. I want to suggest that it's not simply a matter of finding another instance of life so that our understanding of biology is not parochial, as the early exobiologists had hoped, and many of us still do, but also of understanding that the constant deferral of our hopes to have definitively located signs of life elsewhere is a sign of the success of our approach. When Burke created radiobes in the test tube, the idea of having created cell-like forms that grew and subdivided was later discounted because then we thought proteins were more fundamental to life than just cell-like forms. Then proteins weren't central enough for our explanations, and we began to focus on gene-centered theories for the emergence of life. After Arthur Kornberg's synthesis of DNA in the 1960s, not even the laboratory synthesis of the hereditary material was sufficient to hold for long the claim that life had just been or would soon be produced in the test tube, although many journalists tried. As we get ever more knowledgeable about how nature has evolved, and even as we become ever more accomplished at engineering biological systems to obtain outcomes not seen before in nature, what we understand to be central or fundamental or sufficient to our understanding of life continues to shift, as it should. We've never been able to reach that moment where we produce life in the test tube because we keep on improving our understanding of and our ability to manipulate living systems, whether natural or designed. That's a feature, not a bug. This history of ever-constant deferral, that we might someday be able to produce life in the test tube, but can only produce n-1 right now, or that we might someday be able to find life beyond the Earth, but we have only our one local case right now, reminds me of the philosopher Kant's brilliant description of a flamenco dance, the sudden transformation of a strained expectation into nothing. Why is it that what seems most interesting is always at the horizon of our understanding? It's a consistent feature of our efforts to understand the conditions for the emergence and the evolution of life in the universe. And it's that very process of resolution, of deciding what counts as relevant evidence or proof, of shifting the burden of proof, of deciding what approaches to characterizing life we want to use, that's actually at the heart of what origins of life investigators have long done, of what many astrobiologists today do, and of what we do when we gather at meetings like this to refine our thinking. So what might it mean to use this history that I've briefly sketched here as a tool to think through our current situation in astrobiology? If claims for the synthesis of life in vitro have these constant features of constant deferral, how might this better inform our understanding of what we're doing when we look for life in astro? And so here's what I'll suggest. It might challenge any easy assumption that in our quest to understand the history of life on Earth and in the universe, that we will reach clear, unambiguous answers. That the silicon giraffe will walk in front of the Viking camera, that the life detection experiments will give us a clear signal, that we'll find nanobacteria in these Antarctic meteorites from Mars, that we'll be able to confirm a biosignature. It will challenge the idea that we should think of the search for life beyond the Earth in the future perfect. Now, we might find silicon giraffes, but we might not, right? What would it look like if we had a different development of our science? So rather than hoping for a parallel aha moment of discovery, which would be akin to creating life in the test tube, we can acknowledge a more complicated and a gradual building of knowledge and capacity that over the course of time fundamentally transforms what it means to find life. Maybe we'll find more radiobes before we find microbes. The fact that we haven't found life in other elsewheres so far then, and to use Nathaniel's wording here then, isn't a flaw, but a virtue of our ongoing efforts to produce and to refine our knowledge in this compelling area of inquiry. After all, isn't the history of recent biology and astrobiology, in many important respects, the history of the expansion of our understanding of life, of the conditions for its emergence and evolution, and where else we might find it? And I certainly don't need to remind this audience of our studies of extremophiles, of hot springs, of deep sea vents, of finding amino acids in space, of our shifting from a habitable zone idea defined by planet's distance from the star to envisioning the ways in which more localized gravitational processes may provide other sources of energy, which might expand where we would think to look for life, ocean worlds, as we heard about this morning, or our many proposed theories for the emergence of life from ribozymes and RNA worlds, bird-like dinosaurs, and even more provocative claims that we may be thermodynamically downhill from an abiotic world. The history of astrobiology is already one of the most remarkable stories of scientific achievement. But using other histories of biology might help give us a new way to think about our studies of life here on Earth and in the universe as we search for these new natures, both ones that we invent and ones that we discover. And indeed, both today's synthetic biology and astrobiology are fields deeply concerned with developing a comprehensive understanding of the full potential of living systems, with understanding life as it could be. And that's as true today as it was a century ago. Thank you.
Thank you, Luis. Our final speaker for this session will be Sophia Roosth. She is the Frederick S. Danziger Associate Professor in the Department of the History of Sciences at Harvard. She is an author of a number of articles, and her first book is going to be forthcoming called Synthetic: How Life Got Made, and her talk today is entitled Analysis Synthesis. Sophia?
Good afternoon. Bear with me while I get my technology in order. Historians and Ecclesiastes be damned. In the first decades of the 21st century, a number of things are indeed new under the sun. Living things bearing genomes pared down, streamlined, or cobbled together from bits of synthesized DNA scurry, swim, and flourish in test tubes and glass bioreactors. These include creatures as unlikely as viruses named for computer software, bacteria encoding passages of James Joyce, and chimeric yeast buckling under the combined metabolic strain of genes harvested from wormwood, petunias, and Icelandic microbes. My forthcoming book, Synthetic: How Life Got Made, is an anthropological account of synthetic biologists, a group of researchers who manufacture these novel organisms. Now, this new breed of life scientists has received much press attention. I think some of these passages are actually from Carl. And they assembled in the last years of the 20th century. Researchers from computer science and mechanical, electrical, and chemical engineering together resolved that if the aim of biology was to understand how life works, then manufacturing life would yield better theories than would experimentation. They advocated not experiment, but manufacture, not reduction, but construction, not analysis, but synthesis. New biotechnology techniques, notably faster and cheaper DNA sequencing and synthesis, have aided them in these efforts. In addition to techniques and practices borrowed from their home disciplines, they carried with them a rich trove of analogies and metaphors that directed their thinking about how to manufacture life. So life as code or computational logic gates, if you ask a computer scientist, or as circuits, if you talk to an electrical engineer, perhaps as a chemical factory for those coming from chemical engineering. Since I began my research, synthetic biology has grown considerably. Its laboratories now spread across the United States, the UK, Germany, and the Netherlands. And at last count, over 174 American laboratories report currently conducting synthetic biology research. Between 2005 and 2010, when I was doing the brunt of my research, synthetic biological research was funded by over $430 million dispersed by the US federal government alone. So that's not including funding from other countries or from private organizations. So like I said, I've been studying this community since 2005, and in doing so, I participated and observed as people like this, these are some of my primary informants from Ginkgo Biolabs, worked in labs such as this one, this is the ND laboratory, to engineer new forms of life in the laboratory. But to what end do they fashion such a fantastical bestiary? Coaxing new creatures into recombinant life, I argue, is a way for life scientists to make a persuasive argument about life. In short, synthesis now advances analysis. To understand their ambitions, I want to first offer a much abridged history of 20th century biology, some of which you've already heard today, but I want to do this in order to explain precisely what synthetic biology is not. Since the advent of biology as a professional discipline at the end of the 19th century, life science has, for the most part, been experimental. That is, biologists took organisms apart, flaying flesh from limb, in order to understand their structure and function. Beginning in the 1930s, life science came under the sway of molecular biology, which in investigating the 4 organic molecules common to all living things, that is proteins, nucleic acids, lipids, and carbohydrates, stressed above all the unity of all life on Earth. By the start of the Cold War, Jacques Monod's notorious announcement that anything found to be true of E. coli must also be true of elephants had sunk into a kind of generalized common sense. So what I'm trying to suggest here is that for most of 20th century biology, this has been a project of experimentalism that thinks of life as something primarily mechanical and, most importantly, unitary. Life for these molecular biologists was coherent, it was uniform, it was even at times simple, and its essence was reducible to a genetic command line. Life for synthetic biologists still remains thoroughly materialist, molecular, and reductionist. That being said, their approach to studying life departs radically from their predecessors in molecular biology. Take, for example, this following definition that two synthetic biologists gave to their nascent field in the pages of Nature. They write, "Synthetic biologists seek to assemble components that are not natural, therefore synthetic, to generate chemical systems that support Darwinian evolution, therefore biological. By carrying out the assembly in a synthetic way, we hope to understand non-synthetic, that is, natural biology." So they here equate creating with understanding and synthesis with analysis. It is clear that making life for these researchers is not an end in itself, but rather a technique for plumbing life's limits. It is also, as this quotation suggests, a way of querying seemingly common sense terms like natural and unnatural, biological and synthetic. And here I hope you can hear the scare quotes that I'm placing around the word natural. So I'll now home in on a project that MIT bioengineers launched in the very first years of the 21st century, the so-called T7.1 project. The protagonist of this story is the minimal bacteriophage virus, a creature so small that it is arguably only marginally alive. Bearing a slim genome, in 1983, T7 became one of the first living things to be sequenced, as it weighed in at fewer than 40,000 base pairs. Biologists have described such infectious agents as, and these are quotes from recent articles, pseudo-living, or as existing on the very edge of life. And in the image that you're looking at, the T7 phage is what's surrounding that large thing. The very big sphere is a bacterium. My story begins in the mid-1990s. A scruffy Dartmouth grad student named Drew Endy, a former Amtrak employee trained in mechanical engineering, had recently decided to apply his engineering skills to living creatures. 20 years later, Endy would become a larger-than-life charismatic promoter of synthetic biology, but where I begin, he was merely a frustrated grad student facing an intractable problem. His goal was to build a software simulation that would predict the behavior of the T7 virus, which was a workhorse of molecular biology and genetics, and continues to be one of the most comprehensively understood tools of biological experimentation. Building such a computational model on his reasoning should effectively verify the sum total of all of the data that molecular biologists had gathered about bacteriophage since it had first entered research laboratories in the mid-1940s, right? So the idea is that this predictive model is going to encompass all that we experimentally know about the virus. And all Endy needed to do was computationally model the two things that this virus can do. And if you look at this image behind me, it diagrams what those two phases are. That is, once a bacteriophage infects a bacterium, it can either remain burrowed within the bacterium's genetic material, which is what you see in the upper right-hand corner. This is a phase called lysogeny. Or it can replicate itself, burst through the cell's walls, and infect more bacteria. And you see that in the left-hand corner. This is called lysis. Endy's computational model was, however, an abject failure. It was incapable of predicting this very simple behavior of T7. The problem haunted Endy, and he took a postdoc to learn more biology. Endy later recalled in a series of lectures for MIT's Department of Bioengineering, which I attended, that those years were, as he put it, pretty depressing to me, because now I'm coming back to this problem where I want to understand how this thing works, and I want to understand how this thing works when I shuffle up all the genetic elements. And if I've got 72 elements, then I've got 72 factorial permutations, more than the number of protons in the universe, probably. And so it's not clear if I can build out a computer model that's going to let me explore this, that I'm ever going to be able to get traction on this problem. Simmering down his question to its essence, Andy snapped, "What's wrong with the T7 genome?" Now, I'd like to pause here and marvel at what a bizarre question this is for a biologist to ask. What sort of question is it? Is it an ontological question? Is it a normative one? Is it ethical? Wrong to whom? Or wrong by what metric? Here is precisely where the engineer's penchant for synthesis meets biological analysis. By his logic, his own failure was nothing less than a failure of all of experimental discovery-based biology. All the knowledge that had been painstakingly gathered by classical geneticists, molecular biologists, and virologists over the course of 60 years of laboratory experimentation was to him simply not enough to predict the behavior of an organism so simple that it's arguably only marginally alive. Andy's proclamation that T7 was wrong meant for him that the virus was disorganized, that it was cluttered with genetic junk, and we already heard about junk DNA earlier today. It resisted his best efforts to simulate, model, or understand it. So in short, it was badly designed, and so he set about redesigning it. Upon his arrival at MIT, Andy tasked 2 of his grad students with rebuilding the genetic material of the virus itself, in hopes of manufacturing a virus simple enough that it might accord with computational models. And I want to make sure everyone gets this, so I'm going to repeat it. The computational simulation didn't work, so instead of scrapping the model, they're going to modify the virus itself to accord with the model. The very fact that these researchers choose to rebuild the physical virus to conform to a model and not vice versa suggests the lack of a coherent and meaningful substrate for this kind of biological inquiry. Unable to understand how a living thing works, they remade it in order to make it easier to understand. Engineered organisms, as we will see, now function as proofs of concept, demonstrations of accumulated biological knowledge, as well as signs about what about life remains to be discovered. The logic goes like this. The best way to prove a biological theory or learn more about how life works is to build a new organism from scratch. So if I were to put this in Aristotelian terms of logical proof, such thinking is neither deductive nor inductive, or if you're going to follow semiotician Charles Sanders Peirce, it's not even abductive. Rather, this is a logical proof that is being forward constructively. But back at MIT in 2002, this task fell to 2 grad students. The first was Sriram Kusuri, a new doctoral student in bioengineering who had just done a degree in biology from Berkeley, and Leon Chan, who is a senior graduate student in MIT's Department of Biology. And what happened was this. Armed with new biotechnology techniques like faster and cheaper DNA sequencing and synthesis, they set about redesigning the viral genome. Their first step, and you don't have to understand what's happening here, I'll walk you through it. The first step was to take the entire genome and separate overlapping elements from one another. The idea was that these overlapping elements would code for separate proteins, and that these genes should be able to be manipulated independently of one another. The reasoning is that a well-designed genome should be modular. And once they had separated out those overlapping elements, they began to successively delete those genes that had no known function in order to generate a virus that would bear what they considered to be only the minimal number of genes necessary for life. So to clarify, there are 60 genes in bacteriophage T7, 35 as of right now have a known function, and they decided to cut the remaining 25 to see what happens. In their published paper, Kusuri and Chan posed a question to their readers. They said, should we also expect that the design of an evolved organism would be further optimized for the purposes of human understanding and interaction, right? This virus obviously predates humans by a long shot. So should it be optimized for human understanding? Evidence drawn from fields outside biology suggests that the answer is no. After hearing numerous synthetic biologists pose this same question, I began to ruminate on what life has become when it is something amenable to rational design. What sort of rhetorical possibilities are conditioned by this application of design to life? By 2005, MIT grad students had, and here I'm borrowing the terminology of computer scientists, refactored and rebooted the T7.1 virus, which they named T7.1 following software nomenclature. So follow me now into their laboratory to peer at the sort of unlikely creature they had brought into existence. So here you see 4 petri dishes. On the left is an unengineered, that is a wild type of virus, and it's flourishing in the bacterial culture. On the right, you see the engineered T7.1 virus, which has infected the same kind of bacterial culture. Notice that the bacterial plaques bearing the re-engineered virus are puny and sparse. Simply put, T7.1 is much worse than its naturally occurring predecessor at doing precisely what these viruses have evolved to do, which is infect bacteria, replicate itself, burst open the host, repeat. If natural selection is the gradual modification of an organism to be better suited to its environment, then what is good design if better genomes make worse viruses? Before I could ask my interlocutors this question, they asked me. A few months after the bacterial plaques had been grown in the lab, I went out for dinner with Andy and some of his grad students at a restaurant just off of the MIT campus. And as plates of appetizers were cleared away and the wine glasses were refilled, Andy asked me this question, which I had myself been pondering. He said, is there an environment that would naturally give rise to T7.1? This question was a provocation and a test. It was tangling with questions of nature and artifice and evolution and design. And I paused for a moment to think about it before replying, well, yes, it's called a synthetic biology lab. And Andy laughed approvingly, and he says, great, that's exactly what I say when people pose the same question to me. And he continued, and he says, so in nature, right, the wild type one is totally gonna rock. This is a direct quote. Whereas in my lab, this one's gonna survive, meaning this engineered one, but the other one isn't gonna survive. So by synthetic biologist logic, biological fitness is defined not by adaptability, but by comprehensibility. Synthetic biologists, as engineers bending design principles to living form, build living systems that are more suited to human understanding. As biological making converges with biological knowing, or if you want to use $10 words, as techne joins episteme, a well-designed organism is one that is optimized for human understanding, and most evolved organisms just don't pass that comprehensibility test. The rationale for such thinking, I believe, is the circularity built into this merging of making and knowing. That is, composition for synthetic biologists furthers biological understanding, but sometimes composition is also guided by understanding as itself a design principle. So to clarify that, many synthetic biologists quote Richard Feynman, a famous physicist who wrote on his Caltech chalkboard the day of his death, and you can see the quote right in the upper left-hand corner. He said, "What I cannot create, I do not understand." But in the case of T7.1, a more accurate pronouncement would be, I only create what I can understand. Put otherwise, when making new biological things becomes the path by which biology is best understood, then understandable biological things get preferentially made. Exegesis becomes its own evolutionary force. The unlikely lives born by synthetic biologists grow and mutate and metabolize and divide and senesce, yet they also speak eloquently of nature and artifice, of analysis and synthesis, of theories of life and their limits. And so to close, I want to expand outward from this case to speak to synthetic biology more broadly. This approach to knowing life by remaking it inhabits a different sort of epistemic space than the other methods typical of the life sciences. Namely, and as I said earlier, it's not deductive or inductive. It is at times indebted to things like collection, classification, and experimentalism, which are the traditional methods of the life sciences. But it does not neatly align with the kind of practically driven design and manufacture that one associates, for example, with the biotech industry or with pharmaceuticals. Rather, it's something in between. It is interested making, by which I mean making with a question attached. It is a way of theorizing the biological. The objects of this manufacture incarnate and exemplify answers, even as they point toward as yet unasked questions. And in fact, I would further posit that such active interested making is not limited to T7 or even to synthetic biology, but rather is endemic to the natural and social sciences of our moment. Thank you.
Well, thank you, Sophia. With three such rich talks, we have gone way over schedule. And so the plan is we're going to break for lunch now.
This has been a presentation of the Library of Congress. Visit us at loc.gov.